afternoon, everyone. My name is Himabindu Gazula, and I'll be presenting on detection of human pathogens on fresh strawberries. So, strawberry is an important major crop in the United States, and according to some statistics provided by USDA and other agencies, it has been shown that the production and consumption of these strawberries has increased a lot in the past few decades. But as we consume these strawberries raw and without any prior processing, we can be prone to many foodborne illnesses because of some contaminants and pathogens present in them. On that note, even CDC has confirmed, like the Center for Disease Control, even it has confirmed the presence of so many pathogens in such strawberries. In addition to that, so many outbreaks which we have seen in the past few years, like mainly by the E. coli, Salmonella, it has shown that strawberry is one of the main source of contaminants. If you see the pie chart here, it shows that different causative organisms of the food contamination. And if you observe that E. coli, Escherichia coli, Salmonella, and Listeria, they account for more than 50% of these outbreaks. So this research study is mainly based on those three human pathogens which cause serious diseases. So all these findings leads us to work on three proposed objectives mainly. The first one being the identification of these microbial pathogens in the strawberries, and second one being the survival analysis of these bacteria on fresh strawberry surface, and third one, which is a novel method, the antibody-based detection of these pathogens in the strawberry. So moving on to the first objective, which is the identification of these bacteria. We have collected strawberries from the nearby farms, and then we inoculated these bacteria individually into those strawberries, and then we stored them for around 3 to 4 weeks, and later we took the strawberry extracts and did some studies on that. So these identification studies include the direct spreading, where we took some extracts of the strawberry and plated on some selective medium, and the second one being the enrichment method, where we provided some enrichment conditions for the bacterial population to grow. So for these detection methods, we have used LEMV agar for direct spreading and LSD broth for the enrichment of E. coli, whereas we have used XLD agar and lactose broth for direct spreading and enrichment of the Salmonella species. Whereas for Listeria, we have used this LSA, which is a Listeria selective agar, and BLEP for the enrichment. But these identification processes, during this process, we couldn't find any bacterial population. So we had focused on detecting the bacterial antigens in those strawberries. So we had done the immunoblot studies on that. So if you see, so before doing these immunoblot studies, we had done some lyophilization method to remove the impurities and also the excessive salts present in the strawberry extract. And we had used some monoclonal antibodies, commercially available antibodies, and we had detected the E. coli and Listeria bacterial antigens in those strawberry extracts. In addition to that, we had used even the monoclonal antibodies and the polyclonal antibodies to detect those bacterial antigens. So from these studies, we had seen that even though bacterial population is not present, bacteria is dead in those extracts, we could see the bacterial antigens in them. So this has led us to focus on the to study about the survival of those pathogens in the strawberry, which means like how long the bacteria is going to survive, even though the bacterial antigen is present in them. So this has led to the second objective of our study, which is the survival analysis of the pathogens on the strawberry. So for this study, we had done like we had taken like strawberries from organic strawberries and also the non-organic strawberries, and we had provided some conditions like storing them at 4 degrees and also at 25 degrees. Why? Because usually we store the strawberries at 4 degrees, like for the storage, and 25, which is for the retail display, like at markets and public places. That's a temperature we store them. So for these studies, we had got these organic strawberries from the nearby Nancy Edward home farm of Chandler variety, and the non-organic ones from the nearby Kroger's. So for these survival studies, we had done the screening by following some methods. Like first, we had isolated the bacterial cultures, individual bacterial colonies, and then suspended in some buffers. And then we had followed the spot inoculation method, in which around 10 to 30 microliters of this bacteria is inoculated onto the surface of the strawberry. And then we did homogenization by using an instrument called stomacher, and then we took some extracts, and we did some serial dilutions, and then plated on some selective media plates. And by using a colony counter, after plating 24 to 48 hours after plating on selective media, we had done the analysis studies. So coming to this analysis, for the E. coli, as I mentioned before, we have done like 2 trials. The first one in the organic ones and the second one from the non-organic ones. And here, if you see, the x-axis represents the analysis of the bacterial colonies, which means like right from the first day after inoculation, we had observed the colony numbers right on the first day of inoculation, and third day, and on the 5th day of inoculation. And the y-axis represents the total log of the log values, average total log of the colony forming units per strawberry we observed. And if you see that at 4 degrees on the day 1, around log of 9 value is observed, but on the day 3 we had observed around reduction, 1 log reduction was observed, which means around 100 times the bacterial population was decreased, and even the same decrease was observed when compared to the day 3 to day 5. And similar log reductions were observed in the non-organic ones, which we done on the second trial. So for these analysis studies, we had used this MECOG, which is a selective agar medium for the E. coli. And coming to the survival analysis studies of the Listeria, we had used the Listeria selective agar medium to observe these colony numbers. Here, even if you see that in the first trial, from day 1 to day 3, the log value has decreased from 9 to around from 9.2 to around 7.7 like that, which is around 2 log reductions. So here I would say that around 99% of the reduction was observed. And if we observe from day 3 to day 5, around 1 log reduction, which means around 90% reduction was there. And similarly, I could say that same reductions were observed in the second trial. So coming to the survival analysis studies of the Salmonella, we have done these studies by using XLD4 media supplemented with the tergitol. And even here we have seen some reductions right from the day 1 of inoculation to the day 5 of inoculation of this bacterial species on the fresh strawberry. So from these survival analysis studies, we found that there is a significant reduction in the bacterial colonies right from the first day, which means like from the first day of inoculation to the even after 5 days of inoculation. So if we observe like from day 1 to day 3, which is around 48 hours after inoculation, we had found a decrease in the colony numbers of E. coli and Salmonella and Listeria. And yet at 4 degrees and even at 25 degrees, there was also some reduction. So overall, I would say there is around 1 to 2-fold of reduction in the total bacterial population. And it observed even after 96 hours after inoculation. similar reductions were observed, which is around one-fold. So coming to the conclusion, so these studies, from these studies I could say that these bacterial populations, they did not proliferate much on the surface of the strawberry, and it could be because of this complex nature of the strawberry surface, maybe, which is not supporting the growth of this bacteria. And even as we observe from the previous studies, we observed that even though bacteria population is dead, we could still detect the bacterial antigen, which is a causative of the disease. So this can be treated as a risk factor assessment factor for further studies. So coming to the future studies, we are now working on an antibody-based detection method called the dipstick assay, where we are trying to trying to see whether we can detect even a single bacterial cell with that dipstick assay. And the second one, we are trying to do the metagenome sequencing studies, because while doing these sequence studies, we had observed the growth of some other bacteria growing on these selective media plates. So we are trying to know the sequence, know what kind of bacteria is growing along with our bacteria on the selective media plates, and so that we can, if possible, if it is a harmful one, so we want to develop a dipstick assay for that too. So as a part of this project, we had conducted and we will be conducting so many workshops to demonstrate the farmers how to use this dipstick assay to detect those pathogens, and also to encourage them to produce more organic grown strawberries. So these are the references I have used. And this project is funded by Walmart, and even University of Arkansas is also working with us in developing this project. And I'm thankful to Dr. Suping and Dr. Chen and Kathy and everyone from the lab for the support. Thank you.
Would you expect the bacteria to grow at the temperatures that you use in your studies?
Sorry?
Like at 4 degrees and 25 degrees, which is way below their optimal growth temperature.
Sorry, I didn't.
Now, would you expect the bacteria to grow at 4 degrees and 25 degrees when
No,
most
we
of them...
use those conditions because that's
how you store it.
conditions we store, and even storage and also outside. That's
Yeah,
why
but normally you wouldn't expect them to grow anyway.
sometimes if provided some conditions, we can say that.
And what's your reason about thinking the organically grown strawberries? would be
Because
different?
nowadays people are going more for the organic products. So
Yeah, but why? Why would organic growth have anything to do with the growth of bacteria on strawberries?
so you mean why I'm working on organic strawberries now?
Yeah. What about organic growth would retard the growth of bacteria?
I mean, from these studies we got to know that these bacterial population in organic strawberries, the reduction is more, like reduction in the colony number is more when compared to the non-organic ones.
I think the question is why on organic strawberries you think they are more susceptible to bacteria or pathogenic infections compared to the inorganic ones?
Okay, because
Why they are more
organic
susceptible?
means like you don't use all these pesticides and everything, whatever you provide, which like there would be so many causes of contamination when you go for the organic ones. But if you go for the non-organic ones where you follow the conventional methods, you use all these pesticides and insecticides and everything. So there could be chances that whatever the pathogens present in there, they can die by those spraying of everything. But in the organic ones, as you don't use anything, so there could be chances that these bacteria will grow and could cause disease.
So your recommendation is not to eat organic strawberry?
I'm trying to reduce those even though it's there.